CSIR Evolution Chapter 2. 1. The evidences for evolution. Evidences for evolution are strong proof in support for organic evolution, that it is a continuous process which had occurred in past from the time of the origin of life and is still continuing and will shape the diversity of life in future. On small scale there are some evidences of rapid evolutionary changes which can be directly observed. However, we can also observe large-scale evolutionary changes of biological diversity of hundreds of millions of years. We will first look small-scale evolutionary changes by taking examples of natural selection as an evidence of evolution. 2.1 Evidence of evolution on small scale. 2.1.1 The evolution of insecticide-resistant insects. A classic example of natural selection is the evolution of insecticide resistance in hundreds of insect species. Insecticides are poisons used to kill insects that are pests in farmlands, swamps and homes. Examples are DDT, now banned in many countries, and malathion. The poison kills most of the members of the insect population, leaving the resistant individuals to reproduce and their offspring inherit the genes for insecticide resistance. In each generation, the proportion of insecticide-resistant individuals in the insect population increases. The population has adapted to change in its environment. This example of insect adaptation to insecticides highlights two key points about natural selection. First, notice that natural selection is more a process of editing than it is a creative mechanism. An insecticide does not create resistant individuals, but selects for resistant insects that were already present in the population. Second, natural selection works according to time and place. It favors those characteristics in a varying population that fit the current, local environment. Characteristics which are adaptive in one situation may be useless or even detrimental in different circumstances. 2.1.2 The evolution of drug-resistant HIV. Human immunodeficiency virus, HIV, that causes AIDS is a retrovirus having RNA as its genetic material inside the host cell. By using its own reverse transcriptase enzyme, it synthesizes DNA version from its RNA version. The DNA synthesized in the process of reverse transcription is then inserted into the host cell genome, and this condition is called as prophage. In later stages it multiplies by using transcriptional machinery of host cell to synthesize its RNA component. Reverse transcriptase is normally not present in human cells. The reverse transcriptase is the favorite target for anti-HIV drugs. If reverse transcriptase can be inactivated by a drug, the virus is stopped from reproducing without any damaging side effects on the cell. Many drugs have been developed against reverse transcriptase. One large class of these drugs consists of nucleoside inhibitors. A nucleoside is a nucleotide without the phosphate, it is base plus sugar. The drug 3-TC, for example, is a molecule which mimics the cytosine nucleotide of DNA. The HIV's reverse transcriptase will pick up a 3-TC molecule instead of C and insert it into a growing DNA chain. This error terminates further elongation of the DNA and thus blocks reproduction of the HIV. When AIDS patients are treated with 3-TC, initially the HIV population in the human body decreases by a huge amount. But then, within days, 3-TC resistant strains of HIV start to be detected. The drug-resistant HIV then increases in frequency, and within 3 weeks of the start of the drug treatment the population of drug-resistant HIV increases up to 100% in the patient's body. The change, from a viral population that was susceptible to 3-TC to a viral population that was resistant to BTC, is an example of evolution by natural selection. The evolution takes place within a single human body, and is exceptionally rapid relative to most examples of evolution. The evolution of drug resistance can be understood at molecular level. The change from 3TC susceptible HIV to 3TC resistant HIV is achieved by a change in one codon in the gene that codes for reverse transcriptase. The amino acid methionine is changed to one of the 3 other amino acids. The methionine is in part of the reverse transcriptase that interacts with the nucleotides. Due to this change, the 3TC-resistant variety of HIV has a slightly different version of reverse transcriptase that is able to discriminate between the drug and the normal C nucleotide. Members of the resistant HIV population that inherit the gene for this form of the enzyme have no advantage in the absence of 3TC. In fact, they replicate their DNA more slowly than the normal variety of HIV. But once 3TC is added to the environment of these viruses, it becomes a potent force in natural selection, favoring reproduction of the resistant individuals. 2.1.3 Antibiotic resistance among microbes. Superbug. Antibiotics have always been considered one of the wonder discoveries. This is true, but the real wonder is the rise of antibiotic resistance in hospitals and communities with their use. The extraordinary genetic capacities of microbes have benefited from man's overuse of antibiotics to exploit every source of resistance genes and every means of horizontal gene transmission to develop multiple mechanisms of resistance for each and every antibiotic introduced into practice clinically, agriculturally, or otherwise. 2.2 Evidences of large-scale evolutionary changes. We have examined cases of evolution by natural selection that occur rapidly enough to be directly observed. However, the much larger changes of biological diversity occur on a time scale spanning hundreds of millions of years. For such large-scale evolutionary changes, human observation is too short-term to witness the whole history of life, but there are some reliable evidences from biogeography, paleontology, comparative embryology, homologies, analogies, biochemistry, molecular biology, and taxonomy which has witnessed long-term evolutionary changes. Among these evidences, biogeographical and paleontological evidences are considered as the best evidences in favor of evolution. 2.2.1 Evidence from biogeography. The study of geographical distribution of animals and plants in different parts of Earth is called biogeography. It was biogeography, even more than fossils, that first motivated Darwin and Wallace towards evolutionary view of life. Darwin noticed that species in one biogeographic area tend to be relatively similar. Species living in different areas tend to differ more, even if the species occupy a similar ecological niche. Thus, ecologically different species in one area will share similarities that are lacking between ecologically similar species in different areas. This suggests the species in any one area are descended from a common ancestor. Islands are the showcases of biogeographic evidences for evolution. They generally have many species of plants and animals that are endemic. And yet, as Darwin observed when he reassessed his collections from the voyage of the Beagle, most island species are closely related to species from the nearest mainland or neighboring island. This explains why two islands with similar environments in different parts of the world are populated not by closely related species but by species taxonomically affiliated with the plants and animals of the nearest mainland, where the environment is quite different. Island chains or archipelagos are especially interesting in their biogeography. If a species that disperses from a mainland to an island succeeds in its new environment, it may give rise to several new species as populations spread to other islands in the archipelago. The name given to this process, adaptive radiation, signifies the rapid evolution of one or a few forms into many different species that occupy different habitats within a new geographical area. The evolution of Darwin finches on Galapagos Islands, Figure 4, and the marsupial radiation in Australia, Figure 5, are good examples of adaptive radiations. The Galapagos finches were an important clue to Darwin's thinking about the origin of species. These finches are believed to have descended from a single ancestral species that colonized the Galapagos archipelago, America, over a short period of time. The Galapagos finches differ in their beak shape and size. Different species feed on seeds that vary in size and hardness. Beak shapes changed in response to different seed types, and these changes were inherited by subsequent generations by favorable natural selection. Alfred Wegener, the German meteorologist and geophysicist, proposed the theory of continental drift in 1915. His theory proposed that the continents were joined initially and got drifted apart over past 300 million year time. He found the fossils of same species of freshwater reptiles in Africa and South America. It was a major piece of evidence supporting his theory on the continental drift. On such fossil was of Mesosaurus, a small reptile that swam in shallow coastal waters about 275 million years ago. Figure 4, evolutionary tree of Darwin's finches showing beak adaptations of the individual species. Since the Mesosaurus was a freshwater reptile, there was no way it could have swam across the Atlantic Ocean, which has salt water, to both continents. So that meant Africa and South America were at one point joined together. The flightless birds ostrich, rhea, and emu are distributed on different continents. This also suggests that all the continents used to be one single supercontinent, Pangaea, earlier, and the flightless birds were isolated after the breakup of the landmass. Australia is the home of subgroups of mammals, the prototherians and marsupials, figure 5, which are distinct from another group of mammals, the eutherians, that lives elsewhere on Earth. Some of them are also found in New Guinea, which was connected to Australia in more recent geological times. Eutherians are mammals that complete their embryonic development in the uterus, while marsupials are born as embryos and complete their development in an external pouch. A marsupium or pouch is one of the features that characterize marsupials. Although not all have a permanent pouch and a few have none at all. Prototherians are egg-laying mammals like platypus and echidna. Figure 5, adaptive radiation of Australian marsupial mammals showing the many divergent forms that evolved independently of changes occurring among placental mammals on other continents. For example, a forest-dwelling marsupial called the sugary glider is superficially very similar to flying squirrels, eutherians that live in North American forests. These two mammals have adapted the same way of life, but they evolved independently from different ancestors. The sugar glider is distinctly marsupial, much more closely related to kangaroos and other Australian marsupials than to flying squirrels or any other eutherian mammals. The sugar glider is a marsupial not because that is a requirement for its gliding lifestyle, but simply because its ancestors were marsupials. The unique fauna of Australia diversified on that island continent after it became isolated from the land masses on which placental mammals diversified. The resemblance between sugar glider and flying squirrels is an example of what biologists call convergent evolution. Several distinct time periods and different routes are explained for the entrance of marsupials into Australia. 1. According to first hypothesis, during late Jurassic, early therians arrived in Antarctica, Australia, where the marsupials subsequently evolved. 2. Another view suggests that during early to middle Cretaceous, early marsupials arrived in Australia from northern regions and then radiated in isolation. 3. Third hypothesis suggests that during Eocene epoch of Tertiary period, there was chance dispersal of marsupials into Australia. Most accepted hypothesis is that marsupials evolved in North America, found their way to South America, and then into Australia via Antarctica when the southern continents were joined, Gondwana, during Jurassic. In Australia they diversified to fill many niches. 2.2.2 Evidences from fossils. Fossils are remains, traces, or other direct evidence of past life forms. Most fossils form from burial of plants and animals in sediment, soft parts are more often consumed or decomposed but may leave imprints if buried rapidly. Most fossils are embedded in sedimentary rock. weathered particles that provide strata from lower older layers to upper newer layers. Paleontologists study the fossil record based on boundaries between strata, where one mix of fossils gives way to another. Transitional links are intermediate between major groups. At any particular location, sedimentation is not continuous but occurs in intervals. When the sea level changes or lakes and swamps dry up and refill. Even when a region is submerged, the rate of sedimentation and the types of sedimentary particles vary over time. As a result of these different periods of sedimentation, the rock forms in strata. The organic substances of a dead organism buried in sediments usually decay rapidly. However, hard parts that are rich in minerals, such as the shells of many invertebrates and protists and the bones and teeth of vertebrates, may remain as fossils. Rarer than mineralized fossils are those that retain organic material. They are sometimes discovered as thin films pressed between layers of sandstone or shale. For example, paleontologists have discovered plant leaves millions of years old that are still green with chlorophyll and well enough preserved for their organic composition to be analyzed and the ultrastructure of their cells to be explored with the electron microscope. The most common fossilized plant material is pollen, which has a hard organic case that resists degradation. Most of the fossils are not remnants of organisms, but are found as rocks as replicas of organisms. These fossils are formed when a dead organism captured in sediments decays and leaves an empty mold that becomes filled with minerals dissolved in water. The minerals may subsequently crystallize, forming a cast in the shape of the organism. Some fossils are found as traces in form of footprints, animal burrows or some animal activities. These fossils tell paleontologists about how the animals that left the traces fossils lived. For example, dinosaur tracks provide clues about the animal's locomotion. In some cases, entire animal bodies including the soft parts are fossilized. Such fossils are formed only when there is no microbial decomposition. For example, an insect got stuck in a drop of resin from tree about 30 million years ago. The resin eventually hardened into amber. These different types of fossils help biologists to reconstruct the history of life. Isolation between populations encourages the evolutionary process. Researchers rarely found transitional forms in the same place as the original forms. Thus, a complete evolutionary progression of fossils, from most ancient to most modern, has never been found in a single locality. A long-standing misperception of the fossil record of evolution is that fossil species form single lines of descent with unidirectional trends. Such a linear view of evolution is called orthogenesis or anagenesis, and has been rejected by paleontologists as a model of evolutionary change. The reality is much more complex than that, with numerous branching lines of descent and multiple morphologic trends. Figure 6. The fossil record of many species reveals that the history of life can be understood as a densely branching bush with many short branches. Cladogenesis. Figure 6. Comparison of orthogenesis with a branching phylogeny, cladogenesis. Connecting links may represent major adaptive shifts. Of special interest in the history of life are the morphological transitions associated with the major adaptive shifts from water to land and land to air. These major changes in mode of life opened up tremendous new adaptive opportunities for animal life. The transition from water to land was one of the most significant events in animal evolution. Recent paleontological and systematic work has shed new light on this transition. The most primitive amphibian yet known is the late Devonian Ichthyostoga, a tetrapod with a flattened skull and bearing a tail fin. The limbs were until recently poorly known, but new fossil evidence has come to light. The hand, previously unknown, shows that these amphibians possessed 7 to 8 digits. The limbs also had a very limited range of movement, and the animal was not as well adapted to terrestrial locomotion as previously thought. The Rhipodistan fishes are widely considered to have given rise to the amphibians. One small group of late Devonian rhipidistions, the panderichthyids, appears to be closely related to the ichthyostogids. These fishes have flattened skulls very similar to that of the early amphibians. In addition, the anal and dorsal fins are absent, and the tail is very similar to that of ichthyostoga. The lobed pectoral and pelvic fins have bones that homologize with the limb bones of the tetrapods. Whether part of a single direct lineage or not, ichthyostogid amphibians and panderichthyid fishes are clearly transitional forms between class-level taxa. The transition from land to air was give rise to arboreal life. One important discovery in 1861 of a true missing link in this case, an animal that stood approximately midway between reptiles and birds known as Archaeopteryx, figure 7. Archaeopteryx had a number of reptilian features, including teeth and a tail of 21 vertebrae, but also had a number of bird-like features such as wishbone and feathers. It was considered as a probable cousin to the lineage running from reptiles to birds. Figure 7I, the fossil Archaeopteryx found in the Upper Jurassic limestones of Bavaria. B. Restoration of the bird's appearance. The organisms which possess the characters of two different groups are referred to connecting links. Examples of connecting links are shown in Table 1. 2.2.3 Methods to estimate date of fossils. To trace evidences in favor of evolution, fossils are reliable historical data only if we can determine their ages. Here two methods are discussed for determining the age of fossils and where fossils fit on the geological timescale. Relative dating. Relative dating determines the relative order of the past events, without necessarily determining the absolute age. The trapping of the dead organisms in sediments freezes fossils in time. Thus, the fossils in each stratum of the sedimentary rock are a local sample of the organisms that existed at the time that sediment was deposited. Because younger sediments are superimposed upon older ones, thus it tells the relative ages of fossils. Relative dating by biostratigraphy is the preferred method in paleontology. Biostratigraphy is the branch of stratigraphy which focuses on correlating and assigning relative ages of rock strata by using the fossil assemblages contained within them. Usually the aim is correlation, demonstrating that a particular horizon in one geological section represents the same period of time as another horizon at some other section. The fossils are useful because sediments of the same age can look completely different because of local variations in the sedimentary environment. For example, one section might have been made up of clays and marls while another has more chalky limestones, but if the fossil species recorded are similar, the two sediments are likely to have been laid down at the same time. The strata at one location can be correlated with strata at another location by the presence of similar fossils, known as index fossils. The best index fossils for correlating strata that are far apart are the shells of sea animals that were widespread. To work well, the fossils used must be widespread geographically, so that they can occur in many different places. They must also be short-lived as a species, so that the period of time during which they could be incorporated in the sediment is relatively narrow. The longer live the species, the poorer the stratigraphic precision, so fossils that evolve rapidly are favored over forms that evolve much more slowly. Absolute dating. In absolute dating, the age is given in years instead of relative terms such as before and after, early and late. Radiometric dating is the method most often used to determine the ages of rocks and fossils on a scale of absolute time. Fossils contain isotopes of elements that accumulated in the organisms when they were alive. Because each radioactive isotope has a fixed rate of decay, it can be used to date a specimen. An isotope's half-life, the number of years it takes for 50% of the original sample to decay, is unaffected by temperature, pressure, and other environmental variables. One of the most widely used and well-known absolute dating techniques is carbon-14 radioisotope dating, which is used to date organic remains. This is a radiometric technique since it is based on radioactive decay. Carbon-14 is an unstable isotope of normal carbon, 12C, and decays to stable nitrogen-14 with beta decay. The half-life of carbon-14 is 5,730 years. Cosmic radiation entering the Earth's atmosphere produces carbon-14. While an organism is alive, it assimilates the different isotopes of each element in proportions determined by their relative abundances in the environment. Carbon-14 is taken in trace quantities, along with much larger quantities of the more common carbon-12. After an organism dies, it is covered with sediment, and eventually becomes consolidated into a layer of rocks as the sediment is compressed. From the time the organism dies and ceases to assimilate carbon, the amount of carbon-14 relative to carbon-12 in the fossil declines due to radioactive decay, figure 8. The age can be determined by the ratio of the two isotopes to learn how much half-life reductions have occurred since it died. For example, if the ratio of carbon-14 to carbon-12 in the fossil was found to be 1/4 that of a living organism, this fossil would be about 11,460 years old. Figure 8, decay of C14 with half-life of 5,730 years. Because the half-life of carbon-14 is 5,730 years, carbon dating is only reliable about up to 50,000 years. To date older fossils, paleontologists use radioactive isotopes with longer half-lives, Table 2. For example, paleontologists uses potassium-40 whose half-life is 1.3 billion years. And uranium-238, which has a half-life of 4.4 billion years, has been used as a radiometric clock to date fossil-containing rocks of the Cambrian period. These radioactive isotopes used in radiometric techniques are also called as clocks of the rocks. 2.2.4 Evidences from comparative anatomy. There are many forms of evidence for evolution. One of the strongest forms of evidence is comparative anatomy, comparing structural similarities of organisms to determine their evolutionary relationships. Organisms with similar anatomical features are assumed to be relatively closely related evolutionarily, and they are assumed to share a common ancestor. As a result of the study of evolutionary relationships, anatomical similarities and differences are important factors in determining and establishing classification of organisms. Some organisms have anatomical structures that are very similar in embryological development and form, but very different in function. Similarity in characteristics resulting from common ancestry is known as homology, and such anatomical signs of evolution are called as homologous structures. Since these structures are so similar, they indicate an evolutionary relationship and a common ancestor of the species that possess them. A clear example of homologous structures is the forelimbs of mammals. When examined closely, the forelimbs of humans, whales, cats, and bats all are very similar in structure, figure 9. Wings of bats and tetrapod digits and tendrils of vitis and tendrils of pumpkin are best examples of homologous organs. Figure 9, homologous structures, forelimbs of mammals. Each possesses the same number of bones, arranged in almost the same way. While they have different external features and they function in different ways, the embryological development and anatomical similarities in form are striking. By comparing the anatomy of these organisms, scientists have determined that they share a common evolutionary ancestor, and in an evolutionary sense, they are relatively closely related. Other organisms have anatomical structures that function in very similar ways, however, morphologically and developmentally these structures are very different. These are called analogous structures. Since these structures are so different, even though they have the same function, they do not indicate an evolutionary relationship nor that two species share a common ancestor. For example, the wings of a bird and dragonfly both serve the same function, they help the organism to fly. However, when comparing the anatomy of these wings, they are very different. The bird wing has bones inside and is covered with feathers, while the dragonfly wing is missing both of these structures. They are analogous structures. Thus, by comparing the anatomy of these organisms, scientists have determined that birds and dragonflies do not share a common evolutionary ancestor, nor that, in an evolutionary sense, they are closely related. Some other examples of analogous organs are wings of birds and bats, tubers of potatoes and sweet potatoes, fins of fish and flippers of a whale. Analogous structures are evidence that these organisms evolved along separate lines. The evolution of different organisms, or parts of organisms, in such similar directions was called convergent evolution, indicating that selection for similar habitats in different evolutionary lineages could occasionally lead to functionally similar anatomical structures. Some of the most interesting homologous structures are vestigial organs. These structures are anatomical features that are still present in an organism, although often reduced in size, even though they no longer serve a function. When comparing anatomy of two organisms, presence of a structure in one and a related, although vestigial structure in the other is evidence that the organisms share a common evolutionary ancestor and that, in an evolutionary sense, they are relatively closely related. Whales, which evolved from land mammals, have vestigial hind leg bones in their bodies. While they no longer use these bones in their marine habitat, they do indicate that whales share an evolutionary relationship with land mammals. Humans have more than 100 vestigial structures in their bodies. Some of the vestigial structure in humans is given in Figure 10. On a superficial level, vestigial organs may seem to support Lamarck's use and disuse concept, but now it is well known that the effects of an individual's use of body structure are not inherited by that individual's offspring. On the other hand, vestigial organs are evidence of evolution by natural selection. Because it would be wasteful to continue providing blood, nutrients, and space to an organ that no longer has a major function, natural selection would tend to favor individuals with reduced version of those organs, and thereby tend to phase out obsolete structures. Ultimately, structural changes such as the adaptation of the tail as the main propulsive structure and the reduction of the hind limbs in whales involve changes in patterns of gene expression during embryonic development, because the embryonic processes affect the functioning of the adult organism. They are themselves subjected to natural selection. Thus, vestigial organs represent the changes in an organism's embryonic development by natural selection. Comparative anatomy is an important tool that helps determine evolutionary relationships between organisms and whether or not they share common ancestors. However, it is also important evidence for evolution. Anatomical similarities between organisms support the idea that these organisms evolved from a common ancestor. Thus, the fact that all vertebrates have four limbs and gill pouches at some part of their development indicates that evolutionary changes have occurred over time, resulting in the diversity we have today. Figure 10, few vestigial structure found in humans. 22.5, evidences from comparative embryology. Early in the 19th century, von Baer had noticed the remarkable similarity among vertebrate embryos that are quite different from each other as adults. He generalized such observation into a law that early embryos that of related species bear more common features than do later, more specialized developmental stages. Darwin and others therefore considered the early stages of development more conservative or evolutionary stable than the adult stages. Ernst Haeckel, 1834–1919, further developed and popularized this concept into the biogenetic law or recapitulation theory. Ontogeny recapitulates phylogeny. This notion holds that the development of an individual organism, ontogeny, is a replay of the evolutionary history of the species, phylogeny. Closely related organisms go through similar stages in their embryonic development. For example, all vertebrate embryos go through a stage in which they have pharyngeal, gill, arches on their throats, Figure 11. Indeed, at this stage of development, similarities between asterisk fishes, frogs, snakes, birds, humans, and all other vertebrates are much more apparent than differences. As development progresses, the various vertebrates diverge more and more, taking on the distinctive characteristics of their classes. In fish, for example, the gill arches develop into gills. In terrestrial vertebrates, these embryonic structures become modified for other functions, such as the eustachian tubes that connect the middle ear with the throat in humans. Similarly, the human embryo, like the embryo of chick and other advanced vertebrates, goes through the stage of having a two-chambered heart like a fish. The two-chambered heart condition is restored in adult V fishes. Later on the heart becomes three-chambered, and this condition is restored in almost all amphibians and most reptiles. In mammals and birds the heart becomes four-chambered, and this condition is restored in their adult rest forms. Figure 11. Different vertebrate embryos at different stages of development. Note that each of the embryos begins with a similar number of pharyngeal, gill, arches, pouches below the head, and a similar vertebral column. In later stages of development, these and other structures are modified to yield each vertebrate group's characteristic form. Moreover, development of all triploblastic animals starts from zygote undergoes similar changes to form gastrula having three primary germ layers, ectoderm, mesoderm and endoderm, which have same fate in organogenesis. Earlier embryos of different vertebrates resembles in possessing similar structures like gills, slits, notochord, tail, etc. Not this only in the course of development at different stages an embryo looks like the embryo of different phyla from which the given organism has evolved. 2.2.6 Evidences from biochemistry and molecular biology. Almost all living organisms use the same basic biochemicals. DNA, ATP, many identical enzymes, DNA triplet code, 20 amino acids, introns, and hypervariable regions. Prokaryotes, i.e. true bacteria, do not have introns. This points to a long period of time since all living things shared common ancestry. Similarity of biochemistry is explained by descent from common ancestor. DNA-based sequences. Differences in DNA between a number of organisms shows less difference the more closely related they are. For example, 2.5% difference between humans and chimpanzees, but 42% difference between humans and lemurs. Amino acid sequences of cytochrome C show similarity between human and monkey, distance from human to duck and greater distance to candida yeast. Data are understandable assuming humans and chimpanzees share a more recent common ancestor than do humans and lemurs, ducks, or yeast. Similarly, a comparison of the number of amino acid differences in hemoglobin and several vertebrates corroborates evidence from paleontology and comparative anatomy about the evolutionary relationships among these species. Figure 12. A common genetic code is overwhelming evidence that all life is related. Evidently, the language of the genetic code has been passed along through all branches of the tree of life ever since the code's inception in an early life form. Biochemical and molecular biological evidence is generally consistent with anatomical similarity of organisms and has thus added the latest chapter to the evidence that evolution is the basis for the unity and diversity of life. Figure 12. Molecular data and the evolutionary relationships of vertebrates. Branch points in this vertebrate tree are defined by the number of amino acids that differ from a polypeptide of human hemoglobin. The molecular data are compatible with vertebrate relationships deduced from the fossil record and comparative anatomy.